is question number 2 from Unit 3. And in this question, we're given some information about enolase, which is an enzyme involved in glycolysis in 2 different organisms. In one of these organisms, Saccharomyces cerevisiae, let's say, that's an organism that functions best at 37 degrees Celsius as opposed to Chloroflexus aurantiacus, which functions best at 55 degrees Celsius. And it says the researchers compared the activity of purified enolase from 2 organisms by measuring the rate of the reaction in the presence of varying concentrations of substrate and a constant amount of each enzyme at both 37 and 55 degrees Celsius. Okay, and here are the 3 questions. Question A says, depending on the organism, the optimal pH for enolase to catalyze its reaction is between 6.5 and 8.0. Describe how a pH below or above this range is likely to affect enolase and its catalytic ability. Well, an enzyme is a protein, right? So the idea is that outside of that optimum pH range, that protein, aka that enzyme, will likely denature, which means change its conformation. And what we know about proteins, again, is that when a protein's function is determined by its shape, so when its shape changes, so does its function or its ability to function. And so once the enzyme finds itself outside of its optimal range, it will denature and therefore become ineffective. Okay, that's A. B says identify the appropriate negative control the researchers most likely used when measuring the reaction rate in the presence of each organism's enolase. Remember, a negative control is an experimental setup in which you expect to find no activity or that the absence, you set it up so that the independent variable is absent, right? So in this case, if the negative control would be running this experiment in the absence of an enzyme. I know the enzyme isn't the independent variable here, but if you run this experiment in the absence of an enzyme, then you measure the same dependent variable, which in this case is some product of glycolysis. I don't think that that's even spelled out for us. Anyways, you would expect that there would be no significant accumulation of whatever that byproduct of glycolysis is in the absence of the enzyme. Okay, and then that's how you could be more certain that the results you're seeing from the other 2 experimental groups that contain the enzyme at those different temperatures, you could be more sure that those results are in fact due to the presence of the enzyme. Okay, C, the researchers predict that for any particular concentration of substrate, C. aurantiacus reaction is going to be more rapid at 55 than 37 degrees. Why do the researchers think this? Well, that's because the enzyme from that organism is optimized to work in a 55 degrees Celsius environment as indicated here, right? So just like a pH change can denature an enzyme, so too can a temperature change, which you may remember from experiments we did this year. So we would expect that enzyme that came from the organism that thrives in the 55 degree temperature, that enzyme would be create a more rapid reaction. And that is it for that one.